Hello, hi. This is the final lecture on Ectocarpus. We have already done two lectures, one on general account and thallus structure and affinities and habitat ecology etc. of Ectocarpus alga. Then we have discussed in detail asexual reproduction in Ectocarpus alga in a different lecture. Now this is the final lecture on sexual reproduction in Ectocarpus. We have already seen in our previous lectures about the erect branches and prostrate branches in Ectocarpus. Then we have discussed the components within the cell. Then we have discussed as a part of that there are two types of generations. One is sporophytic generation which are diploid. Then there is haploid generation which are gametophyte. Okay. This we have discussed as a part of asexual reproduction. If you have not watched that lecture on asexual reproduction, I strongly recommend you watching those lectures before watching this lecture. Now, we will in this lecture, we'll discuss about sexual reproduction. Before that, let me tell you a few more things which we have already discussed. There are two types of filaments, as I told, sporophytic filament and gametophytic filament. This sporophytic filament, we have seen that in it bears two types of sporangia. One is unilocular sporangia, which is having only one chamber. Within that, there will be meiotic division and around 64 zoospores will be formed. Then in plurilocular sporangia, we have discussed that there are hundreds of cells, around 660 cells are present. From it, mitotic division happens and around 660 cells are formed, and from that diploid spores will be formed, and these diploid spores will be regenerating into or germinating into diploid sporophytic plant. So what we have observed is that only sporophytic or diploid generation participate in the asexual reproduction. Okay, plurilocular sporangia, that is releasing diploid spores, that is germinating into sporophyte. Unilocular sporangia, that is releasing zoospores, that is forming the gametophytic generation. Okay, that is forming the gametophytic generation. And in this, after the gametophytic generation is formed, from here we are going to discuss the sexual reproduction, because gametophytic generation only will be participating in the sexual reproduction by releasing the gametes. Let us move to that. This was the slide that we have discussed as a part of asexual reproduction. You see that this is the diploid sporophytic generation which is bearing the unilocular sporangia and plurilocular sporangia. I told that there may be species with only unilocular sporangia is made in one thallus and plurilocular sporangia is made in a different thallus, or it can also be possible that both type of sporangia are formed on same thallus. So just see the morphology of this sporophyte for once. Now we are going to see the sexual reproduction. Sexual reproduction begins with the haploid gametophytic generation. I told you to observe the morphology of sporophyte in the previous slide. This is the morphology of sporophyte. Do you see the similarity between this sporophytic generation and this gametophytic generation? Both are same to same, right? Only difference is that in that there was a unilocular sporangia present. Even the physics or the morphology of this plurilocular gametangia which is present in this particular gametophyte is same to same as the plurilocular sporangia which was present in the thallus of sporophytic generation. Okay, so this is why we call this particular Ectocarpus life cycle as isomorphic alternation of generation, because the gametophytic the sporophytic generation and the gametophytic generation are looking similar. Okay, now coming with the sexual reproduction cycle, as we already discussed, only this haploid gametophytic filament takes part in the sexual reproduction process. Okay, what will happen is that there are two kinds of reproduction mode which is present in Ectocarpus. One is anisogamous mode of sexual reproduction. One is isogamous. This is isogamous. Then these two are anisogamous mode of sexual reproduction. In the species called Ectocarpus pusillus, we can see that both the fusing gametes are similar morphologically and in size, and in physiologically both are similar. Both morphologically and physiologically, both the gametes are similar in Ectocarpus bacillus. Okay. Now, one thing you can observe is that these particular gametes are formed either from the same gametangium or from two different gametangium that are present in the same thallus. Okay. Now, what will happen after released from the gametangium? Both these gametes will be swimming in the water for some time, and it will be forming a diploid zygote that will be germinating into a diploid sporophyte. After the zygote is formed, no further meiosis, nothing happens. It is directly germinating into a diploid sporophyte because zygote is diploid and the sporophyte is diploid. So zygote will be germinating into a sporophyte plant. That is one kind of reproduction mode, that is isogamous. Now coming to anisogamous mode of reproduction in Ectocarpus, that is actually observed in two types. One is the kind of reproduction in which there are two types of gametes are formed. Okay, there are two types of gametes are formed. One type of gamete is formed from a larger sporangium. Gametangium, larger gametangium, that is called megagametangium. Then another type of gametes are formed from small gametangium, that is microgametangium. Both will be releasing the gametes of different sizes. Megagametangium will be releasing a gamete with a larger size, and microgametangium will be releasing a gamete with smaller size. So both will be fusing, and that will be causing the formation of a diploid zygote. Okay, so such kind of reproduction mode, sexual reproduction mode, in which both the gametes are of different size. Okay, this is known as morphological anisogamy. It is observed in Ectocarpus secundus. Kindly remember the species name because that will add marks to you more than your competitor, because most of the people will fail to remember the species name, etc. So kindly remember this thing, and this will be adding more mark, and it will be making the examiner more happy when he or she is seeing one particular candidate has written the species name correctly. This is the example, right? So that will be giving more marks to you then compared to your competitor. Then coming to, that is morphological anisogamy. Now coming to physiological anisogamy. Physiological anisogamy means what? They are morphologically similar because the gametangium size is similar in both the strains, and you can see that why it is called physiologically dissimilar. This is morphologically similar but physiologically dissimilar. That means physiologically, when you are observing it from the outside, that means both are same, but inside, within that, there is dissimilarity because both these gametes are being formed from different thalli. Okay, this is from one thallus, this is from different thallus. Okay, so because of that, these particular gametes will be showing physiological anisogamy. Morphologically they are similar. In this kind of anisogamy we can observe in species Ectocarpus siliculosus. Okay, siliculosus, what we are seeing after the release of these gametes, let us call it this particular gametangium present in a different thalli called positive strain, then another called as negative strain. This is just for making a difference. Okay, other than that, there is nothing importance in that. Just for identifying one as male gamete and another as female gamete, we are just denoting a different name. Now, since both are morphologically looking same, how to categorize them into female and male gamete? Once it is released, those gametes which are more active, that will be considered as male gamete, and those which are considered as female gamete will be less active. Once released, this particular female gamete will be going and settling in one particular suitable substratum. All the male, one thing you have to observe, both in the case of all the female and male gametes, one thing is common, whether in morphological anisogamy or physiological anisogamy, after release, this particular female gamete will be settling in a suitable substratum, and that will be releasing a volatile substance called serine. Okay, serine. Okay, serine. S-C-R-I-N-I-N-E. Okay, serine. This particular serine is a volatile substance which will be attracting the male gametes, and getting attracted with that particular substance, this particular male gametes will be attaching to the female gametes. So this thing will happen here as well. After the female gamete is released, it will release or secrete serotonin, this particular male gametes will be coming near. Initially it will come near the female gamete, and what will happen with the tensile flagellum, all will get hooked or attached to the female gamete. This process is called clump formation. This clump formation is unique in the world of Ectocarpus. In the alga only this clump formation are observed in Ectocarpus genus. So after clump formation, one of the particular gametes, male gamete, will contract, will shrink its particular flagellum and gets close, very close, and fuse with the female gamete, and that will release cause the formation of diploid zygote, because this is diploid, this is haploid, both is formed, fused to form a diploid zygote. Again, this diploid zygote will be will be forming, germinating into a diploid sporophytic plant. Once it is attached, once the flagellum is contracted and it is attached to the female gamete, the fusion will happen within 20 seconds. So this is the crux of sexual reproductive cycle in Ectocarpus. In the upcoming slides I have attached the notes, but you take care that you pause the video and write the lecture note of my discussion, because in the notes I have given it very briefly, because otherwise it will take many pages and it will take more time. So you can take the lecture note of my session and just refer the notes that are given afterwards, okay? Because everything that I have detailed in this kind of complete detail is not given in the notes. I have just given an overline, just a broad ideas or broad steps only I have given in the notes attached in the upcoming slides, which you can pause the video and write it if you want. But I strongly recommend taking lecture notes. Now coming to this particular overall summary, if you want to hear, it is that in the species of Ectocarpus we can see two different types of filaments. One is called a diploid sporophytic generation and a haploid gametophytic generation. And what will happen is that in the asexual reproduction only the diploid sporophytic generation is taking part, and in the case of sexual reproduction only the haploid gametophytic filament is taking part. What will happen is that this particular haploid gametophytic thalli, that will be forming gametes, okay, that will be forming gametes, and these gametes will be fusing to form zygote. This zygote without any meiosis will be directly germinating to form a sporophytic filament. Now what will happen, this particular sporophytic filament will be having two kinds of sporangia. One is unilocular sporangia, then a plurilocular sporangia. This particular plurilocular sporangia will be forming what kind of spores? It will be forming either meiozospore or mitozospore. Just imagine, under plurilocular sporangia was a diploid sporangia. Within that, actually, mitosis happen. What will happen? Within the plurilocular sporangia, mitosis happen. Then only that liberating zoospores will be diploid, okay? Because from the plurilocular sporangia, mitosis happens and diploid zoospores will be formed, and those diploid zoospores we have seen in the asexual reproduction slide. From the diploid zoospores only, what is formed? The diploid sporophytic generation. And from the unilocular sporangia, what will happen? Meiosis happens. Okay, meiosis happens. Okay, initially it was diploid, then meiosis happens. It will be releasing haploid spores. Okay, these haploid spores will be germinating into haploid gametophytic generation. Okay, so that was the crux of sexual and asexual reproduction in the Ectocarpus species. So in Ectocarpus species, both haploid and diploid thalli keeps on occurring back to back. From the from the haploid gametophytic filament, it will be forming gametes, and that will form a zygote. You see, from the gametophytic generation, gametes will be formed, that will be forming a zygote, and from the zygote sporophytic generation is formed. That is, from the gametophytic generation a zygote is formed, and from the zygote a sporophytic generation is formed. Both this will be happening back to back, and both these sporophytic filament and gametophytic filament are looking similar, and because of that we call it as isomorphic alternation of generation. With this we have completed the Ectocarpus. Now we will see the rest of the brown algae in upcoming lectures. Thank you.